Okay, this is DNA replication by Andrew Kligerman. This is a strand of DNA. Let's take a closer look. These are the nitrogen bases. Adenine is yellow, thymine is green, guanine is purple, cytosine is blue. These are just used as a reference. This is an origin of replication, or a replication bubble. This is the start of DNA replication. There's a leading strand. Every process of DNA replication has to go from 3 to 5, the 3 ending strand to the 5 ending strand. And these DNA strips are anti-parallel, so the opposite side runs in the opposite direction, as you can see from the drawing here. I'll get into detail about the leading and lagging strand later. These are basic proteins that are involved in DNA replication. Topoisomerase holds the DNA molecule ahead so that it doesn't break or bend or twist in the bad direction. Helicase is responsible for the actual breaking of the hydrogen bonds between the nitrogen bases. That splitting is the replication fork that has an arrow pointing to it. The still standing body proteins hold these flailing strands of single DNA out in the air so they don't flop around and hurt something. This is another protein. This is primase. This is responsible for laying a primer down so that when DNA polymerase 3 comes in, it has somewhere to start from so it can start replicating daughter strands. DNA polymerase can only attach nitrogen bases on the 3-carbon sugar, so it always has to replicate from 3 to 5, the 3 to 5 direction. This is an example of the leading strand, because once it starts, it can just keep going on until the very end of the molecule. The black is the parent strand, the blue is the daughter strand. Those have arrows pointing to them. This is how the lagging strand works. This is much more in-depth and complicated. The primer must always go 3 to 5, like before. So since the helicase is unzipping in the opposite direction, it has to make a series of jumps when it lays its primer down to keep up with the helicase molecule, since it can't directly go in that direction. Likewise, the DNA polymerase can only go from 3 to 5 also, so that's why that happens. This is an Okazaki fragment. It was just named that because some Japanese guy found it. And, yep, this is DNA polymerase 1. This is what's responsible for turning the primers back into DNA, actual DNA fragments, like that. The last molecule here is DNA ligase. This is responsible for connecting strands of DNA that have been replaced by from primer with the Okazaki fragments. This is also used in the leading strand in the beginning, so they just attach like that. When there's damage in the nitrogen bases, it's this process called nucleotide excision repair happens. It begins with this protein called nuclease that takes the damaged nitrogen bases and removes it. DNA polymerase replaces those damaged molecules with good molecules, and ligase once again reattaches the new DNA to the old DNA, and you get a good DNA molecule. Okay, the last step, we're going to talk about telomeres. This is a broad view of DNA replication. We'll focus on the end here, and we'll focus on the lagging strand. At the very end of a DNA molecule, for some reason, DNA polymerase cannot replace that last primer with actual DNA. So there's a shortening problem. After so many replications, the DNA will continuously shorten and lose genes. That's why we have telomeres. Telomeres attach to the end and buffer the DNA so the ending DNA can actually get replaced and doesn't get chopped off. These are basically buffers. And one last protein that's involved here is telomerase. This lengthens telomeres. Both telomerase and telomeres are associated with aging, and there's unusually high amounts of telomerase in lobsters. That's why they live for an indefinite amount of time. And that's DNA replication in its total.